Hey everybody, and welcome to Bio 101 Biology Today, and we are moving online as promised. I am doing my lectures as a screen capture. I'm lucky enough to have an iPad and access to an Apple Pencil, which is probably the greatest thing in the world since sliced bread as far as drawing goes. I love this thing. So it's going to be a lot like just using a whiteboard. First up, okay, you know my name, but since this is going to be up on YouTube, my name is Mary Annarella, and I teach at Holyoke Community College. This is Bio 101, Biology Today, and I hope that everybody here is doing all right. I hope that you are in a safe place and that you are comfortable where you are, that you have reliable internet access. For those of you who are perhaps watching this over at a friend's house, maybe because you don't have access at home, please be aware that you can contact the library at HCC. They may still have Chromebooks available, and they have been giving out hotspots to folks. So if you don't have reliable Wi-Fi, they can set you up with a hotspot. So it's basically it's basically like a little mini tower that will give you an LTE signal, and you can tether off of that on any device that you have. Anyhow, we're going to do the best that we can here. First up, this is the lecture that is going to be for March 31st. Let me think. Do I have the right date here? Yeah, that would be Tuesday. So Tuesday, March 31st. So this is basically going to function as Tuesday's lecture. Some of what we talk about today will be on the exam. The exam is going to be on Thursday, April 2nd, on Moodle. Okay, we are not doing in-person classes anymore, so I can't give you an exam in person, so we're going to have to take our exam on Moodle. It will be open note. Obviously, I am not dumb. I know that people are going to be sitting with their notes, so it will be open note, but it will be timed. You will have a limited amount of time to take this. You may take it any time on April 2nd. I have it set up for you. It like opens up on 1 a.m. on Thursday, Eastern Standard Time. And it will close before midnight. So you will have all day to work on that exam. You will have 90 minutes. Yeah, I think that's what I put up there. You will have 90 minutes for the exam, and if you have an IEP, so those with an IEP, that stands for Individualized Education Plan. If you don't have an IEP, if you don't know what an IEP is, you probably don't have one. But if you do have an Individualized Education Plan that gives you an accommodation for taking extra time, you will get double time. But please give me a heads up about that. I sent you all an email. So, oops, come back, Apple Pencil. Check your email, please, because I put all these instructions in that email for you. And if you are an individual who needs extended time, you know, you usually take your exams down at the Kittredge Center, I need to hear from you to make sure that I give you double time for this. If you have additional accommodations, you need to talk with the Office of Student Disabilities, and please work things out with them. If you meet with a tutor on a regular basis, you're going to be meeting them probably through Zoom or something like that. But the extra time accommodation is the only thing that I can give you online right now. Any other accommodations, you know, if someone takes notes for you or something like that, you need to work that out with the Office of Student Disabilities. It's out of my hands. I can't help right there, but I can give you extra time. So you will take that. Okay, so a little bit about the exam. Let's just get this out of the way first. All right. So this is going to be exam 2. And it's on Moodle. You can poke around and find it in our Moodle class. All right, and it has instructions for you there. You will have 90 minutes. Like I said, most of it's going to be multiple choice. Most of the multiple choice have only one answer. But a few have more than one answer. Okay, and it'll tell you, it'll say something like, which of the following, give you that, which of the following are true. There might be a couple of them that are true. And I have it set up so that you could choose, A and C or B and D and the like. But for the most part, most of them have only one answer. Okay, there will be some short answer. That's basically like a fill in the blank thing. Okay. And there will be a couple essays, or what I call long answer, where you'll have to list several things. When you take the exam, you're not going to be able to go back and change your answers. Okay, you have to submit. And this part will be graded automatically. It'll be graded automatically by the system, because like the short answers, the fill in the blanks is just a one-word answer. The essays I need to grade. You need to grade those by hand. So it will take longer. So you're not going to have your full exam 2 grade back, instantaneously. I need to grade those essays, okay? Oh gosh, I think the whole thing is like 70 points or something like that, because, I had to adapt this exam that's meant to be taken in person and not be open note into the Moodle format, and that took a lot of doing. So I just want to make sure that I'm not overloading us all with too many essays. So there are just a couple essays, just a few of these where you have to do some writing. So obviously you're going to be doing some typing, so please make sure you have brushed up on your typing skills and all that. Okay, so what's on the exam? Okay, wherever we left off from exam 1. I am not going to have you pull out your bulleted list right now because things are just very different right now, and we also went off book a good bit. But we left off at the last exam. We had carbs and proteins on exam 1. So this one's going to include DNA, RNA, which are nucleic acids. Yeah. And I just realized I need to reorient this like this. Sorry, my bad. This is not my first rodeo. Why am I acting like a newbie when it comes to doing these lectures online here? Sorry. So we're rotating. Shoot, I gotta start this over. All right, well, after that nice little technical difficulty, let's get back to this. Anyhow, what's on the exam? Nucleic acids, all that stuff about DNA. And you need to know the structure, the function, differences between DNA and RNA, all that. I'm going to make this point a little finer. There we go. All right, so you need to know how those nucleotides are put together. You need to know the parts of a nucleotide. Let's see, and then we got into lipids. Did we not? We did fats, and we worked on the phospholipid. All right, so know the parts of a fat. Know the parts of a phospholipid. So you got to know that a fat is made up of glycerol plus, one to three fatty acids. You need to know that a phospholipid is basically a diglyceride with a phosphate group on that third carbon on the glycerol molecule. And you need to know that the phosphate end is, very polar and likes water. It's hydrophilic, and that those fatty acid chains, those hydrophobic tails, are very hydrophobic. And you need to know how those all float into place to form the phospholipid bilayer. So the cell membrane structure, that is going to be on the exam. All right. Also on the exam are parts of the cell. So not just the membrane, but all those parts that we went through, the nucleus, the mitochondria, the chloroplast if we're talking plant cells, the cytoplasm, the rough endoplasmic reticulum, ribosomes. I'm going to say dogs and cats living together, mass hysteria. That is a quote from Ghostbusters, if you're wondering. Anyhow, so all the parts of the cell and their functions. So the parts and functions are on there. Also, differences between prokaryotic cells and eukaryotic cells will be on there. You need to know the differences between the two of them. And I'm going to ask you a lot of questions like that. We also spent time talking about hand washing. So hand washing technique and how soap works is going to be on the exam. I wasn't just talking about that for my health. I was talking about it for your health too. Why hand washing is so important and how soap just works to remove microbes from our hands. We also talked a little bit about antibiotics. So this is going to be on your exam as well. Antibiotics. We talked about how they target differences between eukaryotic cells, which are human cells. Human cells are eukaryotes, spoiler alert, and prokaryotes, which include bacterial cells. So the whole point of antibiotics, we talked about how the point is to destroy, let's see if I can get a different color in here, destroy bacterial cells, but leave human cells okay, leave them alone. So we talked about how some antibiotics, some target 70S ribosomes, which you already know from your previous notes that prokaryotes have, but eukaryotes don't. And other antibiotics, some target a substance called peptidoglycan, which is present in bacteria only. I misspelled present. Present in bacteria only. Okay, so if you have a bacterial cell, it's got this peptidoglycan layer in it. All right, and let's say, here's the rest of the bacterial cell. Now let's make it have some other grainy things in there. There's our bacterial cell. And let's say we've got this human cell here, and here's our nucleus and a rough endoplasmic reticulum and a bunch of mitochondria in here. And let's put in a Golgi, let's make some vesicles. All right, there we go. If you have an antibiotic that targets and destroys peptidoglycan, this cell will die, but this cell will be okay, right? We want to when we're treating a bacterial infection, we want to kill the bacteria without killing you. So that's generally how they work. And this is where we left off. All right, this next little bit is going to be on the exam. 2. I was talking about viruses, and the coronavirus is one. Let's make it purple. Okay, a virus. I'm just going to draw it like this, and this should be the last thing in your notes here. Okay, a virus is just a protein coat, or it's called a capsid, and that's the capsid there in purple. It's not a phospholipid membrane at all. It's just a protein coat, and inside the protein coat is nucleic acid, DNA or RNA. There are RNA viruses and there are DNA viruses. Oh, let me just put some DNA in there. Okay, some viruses actually do have some sort of lipid surrounding them as well. Coronavirus is one of those, but this is the most basic form of a virus right here. Just a simple protein coat with DNA or RNA inside. So I want us to look at this and talk about why antibiotics don't work against viruses. All right, antibiotics, let's say you have antibiotics are trying to exploit differences between our human cells, which are up here, human cells, and the thing we're trying to kill. Well, antibiotics are designed to kill, to destroy peptidoglycan. So they'll destroy something that's got this, red fluffy stuff that I drew here. And they won't kill anything that doesn't have that red fluffy stuff. This does not have that red fluffy stuff, so no peptidoglycan. Peptidoglycan. No peptidoglycan. So antibiotics that destroy peptidoglycan, cells with peptidoglycan, aren't going to work against viruses. So you can't use antibiotics to destroy viruses. And another thing is that back up here, some antibiotics target the 70S ribosomes. Viruses don't have any ribosomes. Nope, so they have no peptidoglycan, no ribosomes. So if you treat someone that has an infection with a virus with an antibiotic that destroys 70S ribosomes, the virus isn't going to care because they don't have any. So that's why you can't use antibiotics if you have the flu. It's not going to work. You need something called antiviral drugs. And there are a handful of antiviral drugs out there, but they don't all work against all viruses. So that's why we can't just use these basic antivirals to treat COVID-19 infections, because we need to develop an antiviral drug that is specific for a specific virus. And since coronavirus is so new, we don't know enough about it to develop an antiviral treatment for it just yet. There are a few drugs out there that they're working on that work to destroy infections with a similar virus, and they're showing some promise, but that's just all in trials right now. But there are antiviral drugs that are out there. So for example, herpes simplex. Herpes is caused by the herpes simplex virus. There's herpes simplex 1, herpes simplex 2, I think Valtrex is the trade name. So Valtrex is an antiviral drug that can destroy herpes virus, herpes simplex virus. Herpes simplex, one type of herpes simplex can cause just cold sores, fever blisters that people will get on their lips, and the other one causes genital herpes. So Valtrex has been very helpful to people with that type of infection there. But you can't treat a viral infection with antibiotics. They're a whole different thing. So the best thing that we have to to treat a viral infection is to not get it in the first place. So let's go back to purple here. Easier to not get a viral infection in the first place. So we're talking prevention. And that's what staying home is all about, right? Staying home, social distancing, which I really don't like the term social distancing. I mean, we're still being social. We can be social online. We just need to be physically distant. So really it's physical distancing. But we call it social distancing because, I don't know, is that just easier to say? Who knows? But that's why we're staying home. That's why colleges across the country, across the world, have all closed, and why so many people are out of work because so many businesses have had to close. But social distancing will reduce the number of cases that people are getting all at once so that we don't overwhelm the healthcare system and fewer people will die from this. So avoidance is one way. So prevention, physical distancing, stay home. Stay the F home, hashtag, whatever. I know that's going around. And the other one, you've heard of it, and probably almost all of you here have had a preventive step from getting many viral infections, and those are vaccines. We do not yet have a vaccine against coronavirus, although they are working on it, and as that develops, one is in trial. One for COVID-19 is in trials right now, but it's going to be 12 to 18 months before that trial is done and it's available to the rest of us. So vaccination, this is what's going to be the game changer. So we're all staying home so that we can hopefully stay healthy until a vaccine is developed, because vaccines are the single most important tool that we have in our public health toolbox. Vaccines changed life on this planet. I cannot emphasize it enough. Antibiotics, when they were developed in the 1950s, let's go back up here. Antibiotics in the 1950s. Most of them started coming out. People who used to die of strep infections don't die from that anymore. And it was then, in the 50s, that was a turning point. Also in the 50s, it was 1953 that Jonas Salk announced the first modern vaccine against poliovirus. Prior to that, I believe 1951 was the most recent polio outbreak, and something like, I gotta look this up, but 18,000 Americans were disabled by contracting polio. That's the one that FDR had, Franklin Delano Roosevelt, president who was in a wheelchair. He had contracted the poliomyelitis virus and was disabled very rapidly from its infection, because polio virus gets into your nervous system and it damages your nerves. But in 1951, I mean, things were not as dire as they are right now with coronavirus, but things were shut down. I mean, beaches and swimming areas were all closed because that's how transmission happened. And people were terrified. They really, really were. And even then, that was not a pandemic. What we have now is a pandemic, and it's global. So this is really serious stuff, and I'm probably preaching to the choir here because you're all in this biology class and you're all staying home, but I can't emphasize enough to just be as careful as you possibly can and stay home. And we're just hoping for a vaccine right now. So do what you can to support public health. Work from home as much as you can. Wash your hands. Anyhow, we're waiting for a vaccine, and as things for the vaccine develop, I will come back and we will talk more about vaccines and how they actually work. So for now, we're going to move on to new material that will not be on the exam. So maybe I need a new color here. What do we like? I like blue, but I don't think that's going to show up very well. Let's go with this russet color, shall we? Here we go. So this will be new material that will be on the third exam. So this will be for exam 3, not exam 2. Exam 2 is on Thursday, April 2nd. So this new stuff, this will be for exam 3. All right, so we have talked about the cell. Here we go. We've got a nucleus and all this stuff here, and ribosomes and all this. This is looking like a very sad cell. But now we're getting into the part of the course that I call cool stuff cells do. Well, they make energy. They make more of themselves, make more cells. They differentiate. They have different functions. But so before we can talk about those, we need to talk about how things get in and out of the cell. So how substances get in and how substances that get out. To do that, substances have to cross the cell membrane. So this next section is transport across the cell membrane. How things get in and out of the cell. There are two basic ways that things can get in and out of the cell. Right? Things, or two categories. There is what we call passive transport and versus what we call active transport. Active transport and passive transport. Okay, here's the little question of the day. Which one of these two, passive transport or active transport, takes energy, and which one does not take energy? Think about that for a sec while I change my color on my pen and sing the Jeopardy song. Do 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 bum bum ba da 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 da. If you said that active requires energy, you are right. In active transport, it requires energy for a molecule to move across. Cross in or out of the cell. Whereas passive transport does not require energy. No energy required. No energy required. All right. So the first one I want to look at is passive transport. Let's take a look at that. So passive transport across the cell membrane does not take any energy. I am trying to remember if you all even had your lab for movement across the membrane. The example of passive transport across the cell membrane is called the process of diffusion. I misspelled diffusion. There we go. One S, two F's, one S. Diffusion across the cell membrane. If you haven't had that lab, you're going to have it virtually. You'll have a virtual lab. Diffusion. Let's leave a little bit of space here in our notes, but the best way to show you diffusion is that if, this is just kind of classic, if you have a glass of water right here, and let's say I put red dye in it, just squirt a little bit of red food coloring, I'll kind of have this little swirly red blob of dye there in the middle. Now you know that if we let this sit there for, you know, a half hour or an hour or whatever, it is not going to look like this later on. All right, and here's the part in an in-person lecture where I'd ask you all, what's this going to look like if we let it sit around? And one of you is going to go, well, you won't have this red blob anymore. It'll be all spread out. The whole glass will be red. And I'll be like, really? Is it going to be red? And you'll be like, well, it won't be red. It will be pink. And that's because in this blob of red dye here, and I'm pointing at the red dye in the middle here, I'll put some dots here, that you know that a solution of dye is a whole bunch of molecules that are really packed in tightly to each other. So there are all these little blobs of red dye. As this diffuses, these molecules are going to move out here so that they have room. So what we do is what we have is a high concentration of dye in the blob, but out here in the water, we have a low concentration of dye. What diffusion is that it is movement from an area where molecules are highly concentrated, moving out to where they are less concentrated. So diffusion, it is movement of a molecule from an area where it is highly concentrated, and I'm just going to write CONC for concentrated just to save us some writing. So movement of a molecule from an area where it is highly concentrated here in this blob to an area where it is less concentrated. So what these dye molecules here are going to do is they're going to spread out and take up as much space as they can, kind of like people. You can pack a whole bunch of people into a very small space, right? But nobody's really going to like that. But if you give these people a bigger space, aren't we all going to spread out and take up as much individual room as we can? Something like that. It's the same deal with these molecules. Over here, they are packed in wicked tightly in this area here. You give them room to spread out, they will, and that spreading out is called diffusion. The molecules are moving from an area where they are highly concentrated to where they are less concentrated. I'm going to get rid of my people here. I just love that. I love this pencil. It makes this possible. Okay. All right, so I want to talk about something here. This situation over here, I'm pointing here, this situation. Get back to that. The whole idea, the whole situation here being that we have an area of high dye concentration here and low out there in the solvent. This situation is called a concentration gradient. concentration gradient. It is the idea that there is an area of high concentration of some substance, and right next door to it, over here, where I'm dotting things, there is a low concentration next to it. So there's a difference between, oops, I lost my water molecules there. I mean, my glass of water, there we go. Okay. This situation here is a concentration gradient. But over here, in this other one right here, this second glass after it's diffused, there is no longer a concentration gradient. There's no difference in concentration from one molecule over here than it is to this area over here. It's all the same. So we say that the gradient has been equalized. It's been equalized and there is no longer a gradient there. Now think about this. When you squirt dye into a glass of water, you know you don't have to do anything for it to diffuse. Those dye molecules are going to spread out all by themselves, so it takes no energy at all. And I want to talk about why. And it has to do with this concentration gradient thing. And the deal with a concentration gradient is that they're not stable. This here is not a stable situation. Whereas this over here is stable. So I say this like this. I say basically a concentration gradient is not stable. Sometimes I say that nature doesn't like a concentration gradient. Nature doesn't favor it. Nature wants the gradient to be equalized because that's stable. Why that is, I don't know. That is literally one of the laws of physics. That is one of the laws of the universe, okay? And we'll get into those later on too. But it is just a law. A law is just an observation. It is just an observation that a concentration gradient is not stable. Whereas when the gradient has been equalized, this is stable. A very stable situation. And you know you don't have to do anything to get those molecules to move from an area of high concentration to an area of low concentration. It does it by itself. Kind of like if anybody ever, drove to HCC up Mountain Road from East Hampton. Okay, you're going down the backside of Mount Tom when you leave HCC to go into East Hampton, and it is a wicked steep hill. And it's got, this sign at the top of it saying big trucks gear down, and that means so that they don't go careening off the side of that mountain. But it is a really steep drop, and the road's got to zigzag back and forth. Okay, so here at the top of the mountain, you have an area of high elevation. And down at the bottom of the hill, you have an area of low elevation. Or at least lower elevation, right? All right. We don't have to be a rocket scientist to know what would happen if we have a ball at the top of the hill and we just barely nudge it. We know that ball is going to go down easily, right? And it's even going to have all this energy at the bottom. It's going to go quite a ways, right? And it doesn't take any energy for that ball to go down the hill. Just like it doesn't take much gasoline, it doesn't take any gasoline for you to take your car down Mountain Road, right? This ball or this car going down Mountain Road, going from an area of high elevation to low elevation, is like diffusion, going from an area of high concentration to low concentration over here. It's like those dye molecules are going downhill when they move from an area of high concentration to where they are less concentrated. So whereas this is a concentration gradient up here, let me erase that tiny little thing. Erase that there. Over here, this is what I would call an elevation gradient, right? It doesn't take any energy to go downhill. Likewise, up here, this whole thing here is like these dye molecules are going downhill. They are going down a concentration gradient. Here, they're going down an elevation gradient. And you all know it's easier to go down than back up. All right. So I'm going to back up here and add to my definition of passive transport. Let's use a different color here. Passive transport doesn't require any energy because it moves a molecule down a gradient. It's like moving a molecule downhill. All right, so what's this diffusion thing mean for the cell? Well, let's look at that scenario. Okay. So for the cell, all right, let's give yourselves a nucleus here, put some mitochondria in here, some ribosomes and the like. Okay, let's say you breathe in and your alveoli in your healthy lungs will absorb the oxygen that's in the air, and let's make oxygen blue. And the hemoglobin in your red blood cells carries it to, I don't know, a cell in your big toe. Let's say this is a cell in your big toe, and your blood system, your vascular system, basically drops off a whole bunch of oxygen just outside, your cells there. You've got a high concentration of oxygen out here. You have a low concentration of oxygen in the cell. What is that called? It's called a concentration gradient. This is a concentration gradient because you have a high concentration of oxygen outside and a low concentration inside. Nature does not like a concentration gradient. It is not stable. Nature does not want 1, 2, 3, 4, 5, 6, a concentration of 6 out here and a concentration of 0 in here. What would make that even? And this is one point where someone would raise their hand and say, well, 3 and 3. So we're like 3 of these need to move in, and they will diffuse right into your cells. Just like that. Diffusion drives oxygen into your cells, and it does not take any energy to do that. Good thing, huh? That we don't have to work so hard to get oxygen into our cells. It's diffusion that drives those oxygen molecules into your cells. Now we have 3 and 3, and that's a stable situation. Okay. And before someone goes, well, how do the rest of the, oh, shoot, don't do that. Before someone says, well, how do the rest of these get into your cells? Aha, yes, there's a solution for this. What happens is that once oxygen is inside your cells use it up, right? You breathe in oxygen, you breathe out CO2. So once your cells use it up, look, now we have 3 out here and 0 in here, so at least one of those has to move in, right? So that actually keeps that concentration gradient going, the fact that we use up oxygen. So there we go. Likewise, likewise, let's get rid of this oxygen here. Okay, you breathe in oxygen and we breathe out CO2, right? So we're going to end up with a bunch of CO2 inside our cells, right? That's toxic for your cells. It's got to get out. But look at what we have here. We have 1, 2, 3, 4 inside the cell and zip 0 outside the cell. That's a concentration gradient. When you have a high concentration of one substance inside the cell and just on the other side of it you have a lower. So this is a high concentration, this is a low concentration. So 1, 2, 3, 4, 2 of these will diffuse right out. Like that. So now we've got 2 and 2, and that's a stable situation. You're actually going to breathe out the CO2, aren't you? Right? Blow that out, and now look, now we have a concentration of 2 inside and a concentration of 0 outside, so that's a concentration gradient all over again. And so that drives the CO2 out of your body, out of your cells. So that is all done by diffusion, and diffusion is for small molecules. Diffusion. Small molecules can diffuse like oxygen, CO2, water. Water diffuses all the time. There's water outside the cell that can diffuse right in. There's water inside the cell that can diffuse right out. But not every molecule can just simply diffuse in and out of the cell. And that's because the cell let's not do that one. The cell membrane, your phospholipid bilayer, it is what's called a oops, did I misspell that again? Semi-permeable membrane. And that means that only certain molecules are allowed to move in and out by diffusion. So small molecules like water and oxygen and CO2 can, but certain things can't move in and out by diffusion. So for example, let's say we've got a cell here. Here's our cell. I'm not going to draw all the other stuff, all right? Maybe a mitochondria and some grainy looking stuff. There we go. And let's say, I don't know, let's say this is a cell that's lining the inside of your mouth. And let's say someday when things get back to normal again, we'll be actually able to go to a theater to see a movie that's not on Disney Plus or Netflix. And, you know, you go and you buy some popcorn. And when you eat popcorn, you know they salt that stuff like crazy, right? So that stuff's too salty for me. But they do that so that you end up getting thirsty and have to buy all these drinks, right? But it's got a lot of salt on it. And salt is sodium chloride, right? But you know how when you look at like the nutritional information on, say, I don't know, a granola bar or a box of crackers, when it talks about salt content, it generally doesn't talk about the chloride content. It talks about sodium content. So when we're talking salt and having a certain level of salt in the blood and in your cells, we're really concerned about these sodium ions. So let's say you ate a bunch of popcorn, it's really salty, and so you are just bathing the cells lining your mouth with a lot of sodium ions. All right, so we got a high concentration of sodium outside the cell, but a low concentration inside the cell. and that is called a concentration gradient. Is a concentration gradient stable? Nope, it is not stable. Nature does not like 1, 2, 3, 4, 5, 6 out here and 0 in here. And many of you have figured out, yes, that means 3 of these need to move in, right? I'm gonna give myself a little bit of room here. Ah, did not mean to do that. There we go. Give me my cell membrane back. All right, so we've got 1, 2, 3, 4, 5, 6 out here, 0 in here. The cell is freaking out, going, oh my God, I got to equalize this concentration gradient. So 3 of these need, oops, shoot, I'm still on the eraser. 3 of these need to move in. And just as the sodium is like, I gotta equalize this concentration gradient, the cell membrane says, nope. I'm a semi-permeable membrane, and you are not on my list of substances that are allowed to cross in. Go directly to jail. Do not pass go. Do not collect $200. That's sodium ion. Sodium is not allowed to cross the cell membrane by diffusion. It's just not. It's just an observation. Sodium can't cross cell membrane by diffusion. Can't do it. So we're screwed. The cell's screwed here. It's We've got this unstable situation where we have 6 sodium, a concentration of 6 outside the cell and a concentration of zilch inside the cell. What do we do? All right. This problem has a solution. And the solution is in the solution. Okay, the cytoplasm. Remember what I told you that the cytoplasm is mainly made up of? It's mainly made up of water, right? That's why those phospholipid have their polar heads facing the watery interior of the cell and the hydrophobic tails facing outside the cell. But there's also water outside in the exterior of the cell, and that's why we got to get that second layer of phospholipids like that. But you know, the basic thing being there's water inside the cell, there's water outside the cell. You eat a whole bunch of salty popcorn, you are bathing the cells lining your mouth with a whole bunch of sodium ions, and those sodium ions start to freak out because they can't equalize this concentration gradient, and it's just not freaking stable. This problem has a solution, and water is like, hold my beer, I got you. Water will actually exit the cell in an attempt to dilute out the sodium concentration that's out here. Can you see that if out here we add a whole bunch of water, that's going to dilute this sodium concentration out there? So water exits the cells. And what's going to happen if too much water leaves the cell? The cell will shrivel. The cell shrivels. All right. And what's that word for when something loses a lot of water? Dehydrates. It dehydrates. And don't you get thirsty eventually? That sends off a trigger to your brain to tell you, hey, drink something. Go buy an extra Coca-Cola or 7-Up. Whatever, Sprite. Drink something because you lost a whole bunch of water. It's the same deal with plant cells too. plant cells will lose water if you put salt on them. Have you ever tried that? Like, if you're cooking at home, now's a good time to test this out, especially if you have like zucchini, because zucchinis are a fruit. Zucchinis are a vegetable that have a lot of water, and you got to be careful when you cook it or they get really soggy, right? But just slice a zucchini round or a cucumber round, sprinkle a little salt on it, and just watch water bead up on that. You are drawing water out of those plant cells when you put salt on it. So when you put a cell, whether it's a plant cell or a human cell, into a very salty situation where there is a higher sodium concentration outside than inside, that's called putting your cell into a hypertonic solution. So let's write that down. Hypertonic. This means that the sodium ion concentration, and that's what those little brackets mean, it means concentration. The sodium ion concentration outside the cell is higher than the sodium ion concentration inside cell. And what happens is that water exits the cell in an attempt to equalize the gradient caused by sodium. And the cell shrivels and dehydrates. Now this process up here of water equalizing a gradient that it didn't cause, water didn't cause the problem here. Sodium was the one making the problem. You ate a whole bunch of salty popcorn and put your cells in a hypertonic solution, and sodium's trying to equalize the gradient, but it can't. So water's like, I got this. I got you. Water exits the cell, and it's like, hold my beer. I will fix this problem. This process of water diffusing across a semi-permeable membrane to fix the concentration gradient caused by sodium is called osmosis. Let's give that a different color. Osmosis is the diffusion of water across a semi-permeable membrane. I can't spell membrane. In order to equalize the concentration gradient caused by a solute. And in this example, sodium is the solute. Okay, water is the solvent. All right, now sometimes, and in some books I even see this, and it's just wrong. Well, they will say, well, for osmosis, water moves from an area of high water concentration to low water concentration. And I'm like, nope. How can water have a concentration if it is the solvent? That is so misleading. Water is equalizing the gradient caused by sodium. Water doesn't have the problem here. Water's not causing the problem. Sodium's causing the concentration gradient that's not stable, and water's got to fix it. All right? So it is moving from an area of high solute concentration. It's moving to an area of high sodium concentration. So I like this definition. I think it makes a little more sense. The diffusion of water as a solvent across a semi-permeable membrane in order to equalize the concentration caused by a solute. And in this case, it is sodium right there. So water moves to where sodium concentration is higher. And in the case of a hypertonic solution, water will exit the cell because a hypertonic solution has a higher sodium level outside the cell. Now, it's a little different in plants. I'm going to talk about a hypertonic solution in plants. Plant cells. Oh, solution, I'm just abbreviating solution with SOL''N, something like there. Plant cells are a little different in that they have a cell membrane that's very flexible. But outside the cell membrane, they have a rigid cell wall. That's not the same thing. A cell wall is not a cell membrane. It's rigid, whereas the cell membrane is flexible. Flexy. This still looks like an H, whatever. It's flexible. Cell membrane is flexible. Cell wall is rigid. Now, if you put a salty solution onto a plant cell, 1, 2, 3, 4, 5, 6, and it's got a concentration of 6 out here and a concentration of 0 in here, nature doesn't, this is a concentration gradient, nature doesn't like that, so 3 of these are going to want to move in, but the cell membrane is going to say, nope, you don't cross the membrane. You're not allowed because it just is what it is. It is in observation in nature. So water will actually exit the plant cell in an attempt to dilute out the sodium ion concentration out here. That's fine. The cell will dehydrate, but the deal is that this rigid cell wall here is going to stay in place. It's not going to shrivel. It stays in place. So what this will look like, only the cell membrane and the contents will shrivel up. So let me draw what that's going to look like. The contents of the cell membrane will shrivel up like this little bag in here. And all the chloroplasts will just be kind of stuck in here. And the cell, that's the cell membrane plus the contents. Cell contents plus the membrane are all in like what looks like this green bag inside. Whereas the rigid cell wall maintains its rectangular brick structure. Alrighty. So yeah, that's one of the reasons, like in wintertime, you all know how we salt the roads. And we salt the roads to keep it from icing over. The salt makes it harder for the ice for the water to freeze on the roads. But in residential areas, if you're driving around a residential area, sometimes you'll see a sign that says low salt area, and that's, for drivers to exercise more caution because the roads have not been as heavily salted there. And the reason we have these low salt areas is usually because they're residential areas where houses are fairly close to the street and people have shrubs and all kinds of nice plants that are growing, near the road. The salt that we put on the road makes it into the soil, and it can dehydrate plant cells like that, and it can kill plants. So we try not to over salt our roads because it's putting them in a hypertonic solution, and all the plants that are out growing near the road are going to become dehydrated and they can actually die. It can kill the plant. So that's hypertonic for you. Next time we will come back and I'll go through hypotonic and isotonic, and we will take it from there. I hope you have enjoyed our first installment of online biology. Please make sure that you take I will not have a separate lecture up for you for the second because that is test day. So I'll have this up for you. I have already emailed you the link by the time you are watching this, so please make sure that you have. And basically all the stuff from today will be on the third exam, and that will come later in the semester. So I will see you around. Please leave a comment on this video if you have any questions at all. I will check it for the next 48 hours. I'll monitor it. I'm not going to be monitoring this one till the end of the semester, because you're supposed to be watching this now, not later. But go ahead and just post any questions you have in the comments, and I will get back to you there, and that way we can all read the comments, and if someone else has the same question as you, well, then you'll get your question answered even more easily. Okie doke. Oh, one more word. I have made this video unlisted. Okay, it is not public. You're not going to be able to just Google it or do a search for it through YouTube. You're going to need the link for that. I mentioned that in the email to you, which I haven't sent yet, but I will by the time you hear this. So make sure you save that email. If you want to come back and watch that, you're going to need the link for it as well. Anyway, have a great day. Good luck on the exam, and we will talk soon. Bye-bye.